We want to focus now on the basic function of neurons, specifically on their ability to generate action potentials. And we'll look at action potentials from the very beginning to the very end. We'll finish the lecture ultimately by talking about basic types of neurons. So we'll talk about sensory neurons, motor neurons, and interneurons. So let's get started. Here I've got a diagram of our axon end bulb with its presynaptic membrane, our postsynaptic membrane on our next neuron. The gap between them is the synaptic cleft here. Okay? So we're at the end of one neuron, the beginning of another. When our action potential depolarizes the membrane coming down here into the synaptic axon end bulb, then that triggers voltage-gated calcium channels to open up, causing an influx of calcium in the synaptic end bulb. This calcium influx then triggers the following sequence. We have these vesicles in our synaptic end bulb, these little bags, as it were, filled with neurotransmitters. When calcium rushes into the axon end bulb, that triggers these vesicles to begin fusing with the membrane, and they release their neurotransmitters into the synaptic cleft. Those neurotransmitters then diffuse across the synaptic cleft, and they bind to receptor sites on our postsynaptic membrane here. At that time, we also start to see neurotransmitter reuptake pumps pulling the neurotransmitters out of the synaptic cleft again and putting them back into vesicles. Here we see one kind of getting in now, so that they are ready to be used again and again and again. All right. So when we think about what happens here we've got our presynaptic end bulb, our presynaptic membrane, our postsynaptic dendrite, our postsynaptic membrane. What happens then? Well, when we have those neurotransmitters diffusing into the synaptic cleft, binding with the binding sites on the postsynaptic dendritic membrane, that triggers a localized depolarization, which then affects these voltage-sensitive sodium channels. Since there's more sodium, 9 times more sodium, outside the neuron than inside the neuron, there's a chemical concentration gradient that wants to push sodium ions into the membrane, or into the neuron. And so we get that chemical gradient. But we also have an electrical gradient because outside is more positive than inside. And so the positive sodium ions are pulled in by electromagnetic forces as well. So we see then this causes a influx of sodium around those receptor binding sites. Oops, sorry. We have a sodium voltage sensitive channel here. When it's closed, it has both its activation and its inactivation gates closed. It's got a selectivity filter that allows it to let in sodium and not other things. And then it's got an inactivation gate. When we get that local depolarization, the activation gate opens up and we have a sodium influx, rapid sodium influx into our neuron at a local level. After depolarization gets significant, it triggers the inactivation gate to close, and then ultimately the activation gate to close as well when it gets repolarized. Now those diagrams are just schematic diagrams. This is a depiction of the Na1K1 AT phosphase, or the sodium-potassium pump. It generates electrical chemical gradients for sodium and potassium that are vital to animal cells, and in doing so, it exchanges 3 sodium ions for 2 potassium ions across that plasma membrane during each cycle of ATP hydrolysis. So we get increased sodium conductance through the membrane, and this results in sodium influx, which further depolarizes our membrane and triggers more sodium channels to open farther and farther away from the initial event. Eventually, if there's continued depolarization, there'll be significant changes in the membrane potential that goes into the cell body and ultimately at the axon hillock. If membrane potentials at the axon hillock cause depolarization of about 10 millivolts, so say from 65 to 55, that will trigger a spike or an action potential at the axon hillock. So we see localized membrane depolarization causes increased sodium conductance, causing sodium influx, which further fuels membrane depolarization. So if we look at our neuron here, the first thing we see is we get, a graded EPSP. Right? We have a graded change in potential in our postsynaptic membrane here as a result of neurochemical binding to those binding sites on the dendritic branches, causing an influx of sodium. That sodium causes further membrane depolarization, causing further sodium influx, further channels to be opened. If there's significant input into these dendritic branches, it travels down into the cell body and starts to depolarize the cell body as well. If the cell body gets depolarized here at the axon hillock, where our axon leaves the cell body, and that depolarization reaches maybe 10 millivolts, so that we're at negative 55 millivolts, that will trigger an all or none spike that will travel down the axon hillock. That then goes all the way down to the synaptic end bulbs, which then begin that process again with the next neuron. So graded excitatory postsynaptic potential in our dendrites ultimately causes graded change in potential depolarization in the cell body. If it reaches negative 10 millivolts, or we get to that threshold level of the axon hillock, that triggers that all or nothing spike, and that spike then gets conducted down the length of the axon to the synaptic end bulbs to begin its communication with the next neuron.